Hello everyone again. I just want to make this video to demonstrate and to explain how to study for any histological component of for the lab portion of anatomy. You'll notice there's a lot of histological structures that you need to find in your lab manual, and there's a lot of resources that we have remotely for you so you're able to study that. And your guide, as always, will be your lab manual, okay? And I'm going to use the CV and circulatory system as an example for how to study your histology. So, for instance, on this page, there's two different sections. Half the page is dedicated to histology of blood vessels, and the other half is for histology of blood. So just right there, how are we going to study that? How are we going to study the blood vessels in blood using our resources remotely? First of all, we're going to guide ourselves with the lab manual and then go to our APR website. That'll be your first go-to, okay? Always go to the APR website. Then go branch out from there. So we want to find blood vessels. So let's type in vessels, lymphatic vessels, blood vessels. I don't really like that. Let's go to the cardiovascular module. Sounds right. Then up here, okay, look, there's the dissection section. You can do 3D stuff. The 3D part is very laggy, and it's not very good. I don't find it too useful. Maybe later on it will be useful. I don't know. There's animations. There's not too many animations, but if you click on different structures, every once in a while you'll notice a symbol like this over here. Definitely click that and watch it. They're pretty good videos. But we want the imaging stuff, which is pretty cool too. That's more supplementary. But histology, let's click that. So we want this for sure. Okay, we're on the histology section. So great. We're seeing all of the histological slides they have available for us. So this looks like a lot of blood stuff because it matches my blood list in my lab manual. So let's go straight to it. All right. So we're finding some erythrocytes, and they've just been highlighted blue. So those are all the erythrocytes. Tags off down here or tags on. Right, so all these red circles are erythrocytes, or red blood cells. Let's click their tag. So we have to find a function for this. Function, transports oxygen and carbon dioxide to and from tissue of body. You could say transports gases if you wanted to shorten that. Got it. So we found erythrocytes, got the function. Next one is leukocytes. What is a leukocyte? That's going to be confusing if this is the first time learning it. All right, looks like we can't do that. Oh, it says white blood cells right on your lab manual. Okay, they're the white blood cells. But let's look at some of the white blood cells. Okay, this is just showing you different things on this one slide. It's a little confusing on how to go back. So I guess what I would do is I would go to endocrine module, then back to the cardiovascular module, click this little icon. Please, in the comments below, if you know a faster way to do that, let everyone know. It'd be really helpful. Okay, so let's find we just did the erythrocytes. Let's find the neutrophils. We're on high magnification. Okay, so that's a neutrophil. What exactly is it? Let's put the tags on. Always put the tags on. Let's read it. It gives a function, phagocytosis of infectious pathogens, especially bacteria. That's good. It's a good function. About 6 words though. We'll use it. So just recognize the shape at this point. There we go. So this is a pretty large white blood cell compared to an erythrocyte. has a segmented nucleus. I'm noticing. Did it mention that? Description. Typically multi-lobed nucleus. Okay. Granules appear pale pink in blood smears. Okay, I think I saw that. Yeah, pale pink. Gotcha. Let's just put it on the erythrocytes. Non-specific cytoplasmic granules, so it has some granules in there. Okay, so figure that out. Then you go to the eosinophil. Wow, I don't know how to go back. Okay, so let's just do my same strategy. All right, eosinophil. We found that. You would do the same thing, put the tags on, find the function, see how it looks. Then you'd find lymphocytes. There's also lymphocytes on here. You can see the lymphocyte slide. Monocytes are on this list. You have different it looks like it's all high magnification. Then you can read the description of it. It has a kidney-shaped nucleus, it would say. It's a pretty large white blood cell, one of the larger ones. And then it has platelets. So if you can't find the platelet on here, is it on here? It isn't. But on this image, it is. So you would be able to search for platelet up here. Platelet. Cardiovascular section, cool. And it gives you all that contributes to clot formation. That's pretty good. Shows you where the platelets are. They're kind of purpley stained. All right. Now for blood vessels, we got to find all the tunics. So we're in blood right now, but we want large vein because that is a vessel. And look, it has some tunics for us. The tags on. Here's the tunica media, internal elastic lamella, tunica intima. You can go to find some muscular arteries and a vein. Here's a muscular artery. Here's actually a vein. And if you're wondering why is that a vein, well, the diameter of the lumen is like a lot larger. Also, the lumen looks a little bit collapsed. Arteries after death seem to keep their structure. Also, you can see these are not comparable vessels. This one's a little bit larger. Maybe it's maybe this artery went through one more branching point and got a little bit smaller, and this one's a little bit farther back and still a little bit larger. But you can see how the tunics are kind of truncated. There's not as much tissue compared to with the artery, especially in the tunica media portion of it. But in your lab manual, it does have a lot of distinguishing features between arteries and veins that you want to review. Also, on the last page, it explains how arteries and veins look, and that should be able to help you. But again, you can put the tags on. Tunica intima. Innermost layer of vessel wall. Function, barrier to blood solutes. There you go. And just keep looking at all the pictures. And that would cover your vessels. Now, the second most essential thing you want to do after looking at the APR website is to go either to Danielle's anatomy page, which is linked on the 3rd page of your lab manual, and go to the general helpful websites and find all these Graziani videos. This is essential. You have to watch these videos. They're just so good, and they have a lot of pictures in there that are really clear for you. So make sure you review blood vessels, the lymph node, blood and red marrow. Watch them all the way through. That's going to take you like about 45 minutes to watch them. I also have them linked on every section. So here we're on the coelom and viscera and circulatory system resources, and I'll have some videos linked, and I also will make sure that I'm going to link the Graziani videos. Next thing you want to do is look out for any pictures that I've put out. I have pictures from the Histology Atlas. You can either look at them here or you can look at them in your Histology Atlas. You can expand them so you can actually read what's on the page. Wait till this goes back. There we go. I also have pictures straight from the APR website that you can look at and expand. And hopefully you aren't recording a video so your computer's not laggy. And I would say, you know, and your book, of course, your book is a good resource if you have time after studying all of this and you still want to keep studying in that same session, you know, review the book for any histology because it's in there for sure. Okay, guys, any questions? Please put them down in the comments below or email me. My email is on the Canvas website for your instructor. It's just my name, mlindh, M-L-I-N-D-H at santarosa.edu. I'll read the comments as soon as you comment. You know, I have a lot of time on my hands. But this is how you're going to study your histology. Okay. I'll see you in the next video.